My name is Emily Dallas, and I'll be talking about my research, which included an investigation of the effect of forest management on light use efficiency in loblolly pine. In the southeastern U.S., loblolly pine forests are an economically important resource for timber, pulpwood, and potentially biofuel. Subsequently, there is considerable interest in understanding the factors that control the rates of carbon sequestration in loblolly forest. Sunlight is one of several controlling factors of photosynthesis rate, which in turn determines the rate of carbon sequestration. Forest stands with different management plans differ in their crown structures, stem density, and average leaf area index. We lack a complete understanding of how forest management of canopy structure affects the forest's utilization of sunlight for carbon sequestration. In this study, we investigated relationships between canopy light use efficiency and forest management regimes by measuring light transmittance and carbon flux in 3 differently managed loblolly stands in central Virginia. The Briar College Land Atmosphere Research Station is a 33-meter tower and a shed laboratory within a loblolly pine plantation. Forest stands that were planted in 1988, 95, and 99 surround the tower. The 1988 stand was thinned several years ago, but the 95 and 99 stands remain unthinned. To investigate the impacts of different ages and management regimes on the canopy light use efficiency in the 3 stands, a study plot was established within each stand. Within each plot, 4 HOBO radiation sensors were set up a few feet above the ground. These sensors measured the amount of photosynthetically active radiation, which we call PAR, that reached below the canopy. On the tower, a few more LICOR PAR sensors measure the amount of light that reaches the top of the canopy, light that is reflected off of the top of the canopy, light that reaches the bottom of the canopy, and the amount of light that is reflected off of the ground. We also calculated the absorbed PAR, which is the amount of light that the trees absorb due to photosynthesis. We accomplished this by subtracting the light that reflected off of the canopy and the light that was transmitted, and adding the amount that was reflected off of the ground. We assumed that each plot had the same albedo as the area that is measured by the series of PAR sensors on the tower. In analyzing the fraction of absorbed PAR, in Figure 6, we see that the 95 stand absorbs the largest fraction of light. 99 and 88 stands are not significantly different from each other. Light use efficiency is the measurement of how much photosynthesis the forest is able to do per unit of absorbed PAR. In order to calculate this for each plot, we measured the net ecosystem carbon exchange using the LICOR LI 7200 enclosed path infrared gas analyzer on the tower. We used a known relationship between net ecosystem carbon exchange, ecosystem respiration, and air temperature to calculate the gross primary productivity. We separated all of the data by wind direction according to the location of each stand relative to the tower. This allowed us to analyze the carbon flux that was coming from each stand. Light absorption data from each stand was only considered in the calculations when the air samples on the tower were coming from the direction of that stand. By comparing primary productivity to absorbed PAR as seen in Figure 7, we determined the light use efficiency of each plot. We found that the stand that was planted in 95 seems to sequester the most carbon per photon. Therefore, the 95 stand seems to be the most efficient. This might suggest that the 95 plot not only absorbs the most light, but also uses the light that it absorbs more efficiently than the other stands. The 88 stand seems to be the least efficient. The 99 stand did not have statistically different results from either of the other 2 plots. Statistical analyses of all of these results were done via ACO-NOVA. Though it seems like the 95 plot is more efficient than either of the other 2 stands, we cannot make any definitive conclusions about optimal forest age or management regime for carbon sequestration based on this study. Upon further investigation, we found that when the wind blew from the direction of the 1995 plot, the air tended to have a significantly higher relative humidity than when it blew from the other 2 directions, as seen in Figure 8. When air has higher relative humidity, plants are able to open their stomata further and do more photosynthesis. Therefore, on days when the air comes from the 95 direction, all of the trees in the forest are probably more efficient, which we don't see in the results since we separated our data based on airflow direction. In the future, similar research should be conducted in a study area that has differently managed stands of trees that are the same age. This would allow us to know how management techniques alone affect light use efficiency and carbon sequestration. This study was conducted over a 3-month time period from September to December of 2015. As we collect more data at SBC-LARS, we will be able to generate more conclusive results. I hope that this presentation has increased your awareness and understanding of the impacts of forest management on carbon sequestration. At this time, I'd like to thank and acknowledge my research mentors, the SBC-LARS research team, the Jeffress Trust for Interdisciplinary Research, the Global Change Center at Virginia Tech, the Fralin Life Science Institute at Virginia Tech, and the Department of Environmental Science at Briar College.